design and validation of the in vitro digestion system. To reproduce the digestive process under controlled conditions and evaluate nutrient release, we developed a 5-stage static in vitro digestion model designed to simulate the physiological transit of food in Octopus Maya. The system uses gastric juices extracted from different sections of the digestive tract: anterior salivary glands, crop, stomach, caecum, and digestive gland. The protocol is carried out at room temperature, approximately 24 degrees Celsius, and consists of a sequential 5-step digestion. Step 1. Mix 10 mg of the ingredient or formulation to be analyzed with 10 microliters of gastric juice from the anterior salivary glands, and incubate for 5 minutes. Step 2. Transfer 10 microliters of this mixture to a new tube. Add 10 microliters of gastric juice from the crop, and incubate for 15 minutes. Step 3. Repeat the process using gastric juice from the stomach and incubate for 25 minutes. Step 4. Add gastric juice from the caecum and incubate for 45 minutes. Step 5. Finally, add gastric juice from the digestive gland and incubate for 30 minutes. After the final incubation, a 10 microliters aliquot is taken to quantify proteins, cholesterol, and acylglycerides using the Bradford assay. This model not only reproduces the natural enzymatic dynamics but also showed a high correlation with in vivo assay results, validating its use as a reliable, cost-effective, and ethical tool for assessing digestibility and nutrient bioavailability. Why do we use the digestive gland as a reference? Enzymatic measurements were focused on the digestive gland, as it is the central organ responsible for the production, secretion, and regulation of proteolytic enzymes. The analysis of the digestive gland gastric juice allowed us to quantify the activities of cathepsins B, H, and L, which proved to be the main enzymes responsible for protein hydrolysis. Although kinetic characterization revealed that alkaline proteases exhibit lower activation energy, and thus higher catalytic efficiency, our study focused on acidic cathepsins, as they dominate within the physiological environment of the octopus. The initial protein digestion in Omaya occurs under acidic conditions, particularly in the digestive gland, where these cathepsins display their maximum activity. Therefore, their characterization represents the most relevant starting point for understanding the natural digestive physiology of the species. Why we evaluated soluble protein. One of the first aspects we addressed was measuring the soluble protein in the experimental ingredients. This fraction represents the portion of proteins effectively available for hydrolysis by digestive enzymes, as opposed to the total protein content, which includes structures that are inaccessible or resistant to digestion. In the context of Omaya nutrition, soluble protein thus becomes a key indicator of ingredient quality and its potential utilization during the digestive process. Together, these results establish a functional framework for understanding protein digestion in Octopus Maya. Cathepsins B, H, and L emerge as the predominant endogenous enzymes. Cultivable bacteria contribute complementary enzymes with potential applications, and the in vitro system provides a robust experimental platform for testing ingredients and formulations under reproducible conditions. This integration of enzymatic biochemistry with applied microbiology represents a significant step toward developing sustainable diets tailored to the digestive physiology of cephalopods, thereby strengthening the foundations for more efficient and responsible aquaculture.